Okay, guys, I'm going to show you how to do that Hardy-Weinberg new lab, which is the mathematical modeling of what would happen in a specific population to the genotypic frequencies based on starting allele frequencies, assuming that the population is in Hardy-Weinberg equilibrium. So what this is all about is understanding how in biology we use mathematical models to predict biological phenomenon and predict it. And this equation is seeing what would happen if a population was not evolving and to see if the allele frequencies do in fact change compared to what we expect to see, that it actually is evolving. I'm going to go fast because this is a screencast and you guys can go back as many times as you need to. We're going to use Excel for this case, by the way. You can find all the materials right here under the class. I put them under the Hardy-Weinberg screencast extra credit. This is the PDF file that I have open right here that I'm going to be referring to, and then you can either use Screencast-O-Matic, which is a web page, or a Google extension that I'm using, which is called Screencastify, in order to do your screencast. All right, so let's first start off. Oops, that's an actual completed. That's what it's going to look like when we're done. Okay, so let's start from the beginning. First thing you're going to do is you can read through, but we're going to start the actual production is on page 6, which says S30 on the bottom. But this is the PDF. It's 16 pages PDF. This is on the 6th PDF page. And you can start seeing that this is where we're actually going to start producing our spreadsheet. And the first thing we're going to do is on D2, it asks us to put a starting allele frequency. Now, it tells you D2, right? Now, stick with that because every other figure or cell that they refer to, like G5 or F5 or E5, is based on the fact and assuming that you actually started in D2. Now, what you're going to start with, this is the p-value for Hardy-Weinberg, and everybody should know that the p-value is the dominant allele frequency, which is represented by A. Now, that you can't read that, so the formatting that you want to do is highlight those back cells and hit merge, and that places it right there nice and even. Now, the Q value, right, would represent the recessive allele frequency in a given population. Now, we're not going to use little a because Excel cannot determine the difference between a capital letter and a little lowercase letter. So if you put big A or little a, it just thinks that that's the exact same parameter. That's the exact same variable, which is A. So if I want to have one variable represent my dominant allele frequency and another variable represent my recessive allele frequency, I need to have those two things be different in the eyes of Excel. And so we're going to use the recessive allele frequency, and we're going to call that B. So B is the same as like lowercase a. And once again, I'm going to format that. Oops. We're going to format that so that it looks nice. We're going to merge. Now, this cell right here, well, you know, if it's 0.5 is P, then the Q is going to be 0.52, but I want to create a mathematical problem that automatically does that for me. And most of you guys might be familiar with Excel, and you know that if you start by putting equals inside of a cell, everything after that is going to be your function values or the part of the equation that you're creating based on whatever math you're trying to make that cell do. Well, if it's 0.5, we know that P plus Q equals 1. So to calculate whatever Q is, you just take 1 minus whatever our P value is. In this case, it's whatever we put into D2. So you can either type in D2 or just click on D2 and it should calculate it for you. So let's test that. So if my P value is 0.6, right, then what's 1 minus 0.6? It should be 0.4. And that's exactly what that cell calculated. So there it is. It automatically changes it to 0.4. So let's go and put everything back to 0.5. Now, you should probably highlight this and color code it just so you can kind of separate everything because that's what they're asking you to do. Okay, now we want to be able to understand what the rand function does. When you type in equals rand, okay, and just hit enter, what you'll notice is that that cell generates a random number between 0 and 1. And so if you pull it down, each one of those cells is generating a random number between 0 and 1. This is important in understanding how we're going to be able to select which allele pops up when going through sexual reproduction or the creation of a gamete. Now, if there is 60% of all the alleles in a gene population is big A, then there's a 60% chance that the sperm or eggs that are created is going to contain a big A in it. If 40% of those alleles in a specific gene pool are little a, or in this case B, the recessive allele, then 40% of all the gametes that are created are going to be B, or in this case the recessive little a, or in this case B. So we need to be able to create a model that does that correctly. So we're going to need to use this random function. So the first thing we're going to do, if you go back to the instructions, now that we've already had that done, is you're going to create this. If a random number is less than or equal to whatever the value is in D2, then you will place the letter A. But if it's not, comma, then you're going to put the letter B. That's what we're going to put in those cells. We're going to do this on E5, and this is going to tell us how we generate our gametes. And so if you copy and paste, it will not work. So you need to actually refer back to that and plug that in exactly in E5. So equals, if there's a random number, right? If the random number generated, okay, is, like we say, less than or equal to whatever the value is, oops, less than or equal to whatever the value is in D2. In order to do that, you have to put $2. Then you're going to put A, comma. If not, you're going to put B. Okay, and then close parentheses. Now, what that tells us is that right now, if the value is 0.5 or below, you're going to put A. If it's above that, you're going to put B. So there's a 50-50 chance that you're going to put that, then it's going to select A or B. But let's change it and see if you understand what's going on here. Now, in this case, right, if you put 0.6, right, then that means if the random number that is generated is 0.6 or 0.5 or 0.4 or 0.3, 0.6 or below, right, which is 60% of all the random numbers that will be generated, then it's going to pull in and plug in the letter A into that cell. But if it's anything else, anything above 0.6, right, which is 0.7, 0.8, 0.9, or 1.0, which represents the 40% or the allele frequency for the recessive allele, it's going to plug in the letter B, okay? And so that's what we see here are the random gametes that are generated in the population. Now, you can, once you've created that formula in there, oops, once you've created that formula, I think I went a little too far ahead in dragging this down, right? You're going to want to drag this down to, let's say, 20 generations. And so we'll take this down to 25. Now, notice that I had to put my cross in that little square in the bottom right-hand corner in order to apply the same function to all of the cells below it. Now, what these things represent are our gametes, okay? And so these are our gametes. I have to do the same thing for both of these columns. This is going to be if, okay, a random number, okay, is less than or equal to, oops, less than or equal to the value that's in $2. You can't just click the cell, okay? Then I'll put A, okay? That's what the quotation marks mean. Say type A, that's what you're going to put into the cell. If not, which is the comma, you're going to put B. If that statement is not true, then you'll put B, okay? And so here we go. These are the random gametes that are generated. So 20 individuals will produce a gamete. In this case, it would be like the male sperm. And then another 20 individuals will produce these gametes in their eggs. Okay, so these are the gametes that we've created based on the allele frequencies. We can color code that. Let's make that, I don't know, whatever color that is. Now I want to combine those gametes. So we're going to actually have sex, and then we're going to make the zygote. Okay. And so what I want to do is another function, right? We've already done this now. We've pulled it down, okay, and we've color-coded it. This concatenate function, which is just combining the two things that are in each cell. And so go back to that. It's pretty simple. This is probably the easiest of all the functions. We're going to go concatenate, and then whatever is in that cell, comma, which is E5, and that cell, which is F5, close parentheses, and then look. There it is. And we're going to apply that all the way down. Now, notice that we have AB and BA. Later on, we're going to have to distinguish the fact that those two things are not different. They're, in fact, the same. They're both heterozygotes. Otherwise, when we add up and calculate genotypic frequencies, we're going to have four categories. We'll have like homozygous dominant, big A. We'll have homozygous recessive, BB. And then we'll have AB and BA as if they're different, but they're not. They're heterozygote. They're one of each, so they need to fall into the same category when we calculate that. So let's color code our zygotes. We've just created our F1 generation ultimately. Now what we're going to do is, now let's write down our genotypes that have been created. We're going to count up how many genotypes. And notice every time I hit F9, look at this, bam, 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 bam. It just applies the same formula. Now this is if we have 0.6 and 0.4. I want to go back and make sure that we stay with 0.5. It's going to do some math. It's pretty much easy for us to understand. Big A, B, all right, and big B. Okay, I've calculated my, I'm going to merge that so it's in the middle, nice and pretty. I like all my letters to be centered because I'm paranoid. So how am I going to calculate or count? How am I going to tally up all the big A's that are in the population, all the heterozygotes, and then all the BBs or the homozygous recessives? Well, whatever this is, I want to count it in any one of these three categories. So that's where the next formula comes in. So if the zygote, right, if the zygote, which is what is in G5, is big A, then I want to put a number 1 in that cell, which counts it as number 1. So let's go back there, and I'm going to go now, remember, equals. If, right, this cell, okay, is big A, okay, then I'm going to put a 1. If not, I'm going to put a 0. Close it. Oops. I think I'm off by a comma. I don't need the comma. Okay. No comma after G5. There we go. Oops. If G5... Oh, equals. If G5 equals big A, then put... There we go. All right, so since it's BA, that should be a 0. Well, we need to do the same thing for if it's BB, right? So equals if G5, which is that right there, right? All right. If, there we go, double click. If G5 equals, quotation marks, BB, quotation marks, right? That's comma. Then you put a 1. If not, you put a 0, close quotation, bam. Now this one, we have to take into consideration that if G5 is AB, put a 1, or also if G5 is BA, put a 1. If not, then put a 0. So that's where you get into the most complicated formula of this entire worksheet or lab, which is this. And in English, that states, if what's in the zygote cell, which is G5, is an AB, you're going to count that as a 1 under the heterozygote genotype column. If G5 is BA, which is the same thing, then you're also going to count it under the heterozygote column and put a 1. If not, this last column, you're going to put a 0. And then you close it off with 3 parentheses. So let's see if Mr. Vieira can actually look at that and do that without having to refer back to his spreadsheet. Okay. If G5 equals AB, then put a 1, comma, if G5 equals BA, also put a 1. Oops. We need a parentheses right here. Also put a 1. If not, put a 0. We got to close them off. 1, 2, 3. And does it do it? Yes, it does. Okay, see? So since it's big A, we have a 1 only in the big A column. Now I want to apply that formula all the way down here. I want to apply that formula all the way down here. And I want to apply that formula all the way down here. Okay, now I want to, I like to put little lines to kind of separate things. Maybe. Now I want to tally these up. Little line. So now I want the sums of all the big A's. So we're going to count up, okay? In the population of, let's say, this 40 individuals, 20 of the males and then 20 of the females, however you want to do it. One's producing these gametes, the other one producing these. Combining, forming this population of 20 new individuals. How many of those 20 new individuals were big A? Well, how many would we expect to be big A? Doing Hardy-Weinberg equilibrium, right? If you start off with P equals 0.5 and Q equals 0.5, well, this represents P squared. So P squared should be 0.25. So 25% of 20, right? We should expect 5 individuals here. And if you do 2PQ, which is this middle one, we should expect 2PQ, if you plug it in with 0.5 and 0.5, we should expect 10 individuals. And then with these, we should expect another 5 individuals because it's 25% or 0.25 of the total 20. So let's find out if that's actually what we get, because that's our expected values. Let's see what our observed values are. Right off the bat, we get 2, not 5. 13. Every time I hit enter or function, it recalculates everything. Okay, so here we go. Instead of, I should see 5, 10, 5, but I end up getting 7, 6, 8. Now, it's really hard to look at this as numbers, so let's turn this into a graph. Let's highlight the actual values that I want to graph. These are the different genotypes. I hold control, and then this is the labels that I want for them, okay? So then let go of control. You're going to insert. I want a bar graph. And when you do that, it automatically makes a bar graph. Now you can go and add chart elements and then title the axes. So the horizontal axes, oops, let me undo that. Okay, sorry, the axes titles. The horizontal, those are the genotypes. Right? Those are the genotypes. This is big A, right? The vertical, right, axes, vertical, that's the what? That's the number of individuals, whether these are birds or plants, whatever, that contain that genotype. Okay, the chart title, right, would be, I mean, genotypic frequency, but it's not really frequency, it's actually the number of individuals that are in each genotype, but from that you can calculate genotypic frequency. So we'll just put genotypic frequency, because these bars do represent the frequency proportionally. Now you shouldn't see this. You should see half, double, half. You should see 0.25, 5, 0.25. So if it was 20 individuals, you should see 5 individuals here, 10 individuals here, and 5 individuals here. So let's just keep on. There we go. We see there, that's more of the graph that we should expect to see, but it's not exactly. Then we hit F5 again, F9. And we can see every time we recalculate that we get different genotypic frequencies. So this would actually be the number of individual genotypes. And then you can do like a little equal arrow. And then I can do the same thing that I was doing with merging. And that's going to follow around here. But I also want to know what is the number of alleles, because I want to calculate allele frequency. Yeah, give me a minute. Go ahead real quick. All right. So we're going to calculate the number of alleles. So I want to know in a mathematical formula that you can get the A allele and the B allele. Okay. So we're going to have to do a mathematical formula that does that. So what you'll see is you need to use the information in these cells right here to calculate that. So I'm going to stop there because I want you guys to be able to figure that out. I want you to be able to use these cells to plug in equal to figure out what type of math formula would calculate the number of A alleles and the number of B alleles. And then also I want you to figure out what are the allele frequencies. Right? Okay, so that is right here. What is P? How would you calculate P? And what is Q? So you need to figure out what is going to go in those cells that would automatically calculate that. Okay. And also you have to ask yourself, why is it that every time I hit F9, I almost see this evolution that goes on, this change in allele frequencies that goes on every time I hit F9. And that's something that we can talk about in class. But once you've done this, you've ultimately created the basis of your spreadsheet. You can then plug in the formula here that will calculate the number of alleles and the allele frequency. And then you can copy and paste this multiple times and plug in new allele frequencies to see what you would get in the F1, F2, F3, F4 generation. So you can just copy and then paste. And then once you've calculated the allele frequencies from this generation, you can just go in here, right, and plug in new allele frequencies that were calculated from the previous generation, and then you can continually see generation after generation after generation, which would be like a spreadsheet or digital representation or simulation of what we did in the old school Hardy-Weinberg Lab with the cards. I hope that clarifies how to do the Hardy-Weinberg Lab. Thanks for watching.